There's also the concept of multi-target drugs. So in order to understand multi-target drugs, let's look at what combination therapy is. So combination therapy is used when typically in several diseases such as tuberculosis, HIV and cancer. And the reason it is done is that we give multiple drugs to the patient. Okay. So this is normally done because what happens is that it can have these drugs, since they have different targets, they can form some sort of a very strong arsenal against the bacterium. And since it is hitting multiple targets, it is unlikely that it will be able to survive this onslaught. Okay. So that is the rationale for using combination therapy. But one of the disadvantages of combination therapy is that there are a number of different medications and they are supposed to be given at different dose regimens. So it is very difficult to follow this over a period of time, because we discussed earlier that each drug has its own pharmacokinetic profile. So therefore, there are benefits in designing a single drug that can act selectively against different targets. Okay. So these are known as multi-targeted directed ligands or multi-targeted drugs. So there are a number of research projects that are now underway to discover new drugs with a defined profile of activity against a range of specific targets. For example, one may try to find out a drug that has an agonist activity for one receptor subtype and an antagonist activity against another. So this compound olanzapine is a drug that binds to more than a dozen receptors for serotonin, dopamine, muscarine, noradrenaline and histamine. So usually this kind of drug is completely unacceptable for drug discovery. But what is the reality is that this drug has been highly effective in the treatment of schizophrenia, right? And this has been attributed to its ability to block both the serotonin and the dopamine receptor. So these drugs, which interact with a large range of targets, are called as promiscuous ligands or sometimes as dirty drugs. But this helps us to treat this disease, and so therefore, if one could sort of restrict the number of targets for this drug to 2 or 3 rather than a dozen, then it is possible that we can achieve the same result. So such compounds would be great starting points for the design of multi-targeted drugs. So these are the aspects that we need to consider when we are looking at what kind of a disease that we want to hit, what is the selectivity that we want to achieve, and so on and so forth. But once we get into the program, we need to be able to test molecules. Okay. So for this, we need to be able to have what is known as a bioassay. Okay. Assay is nothing but a test system, and so we need to be able to achieve the right bioassay. And this, as we will figure out, is very crucial to the success of a drug research program. So there are a number of challenges in designing the right kind of bioassay. The test should be simple, it should be quick, it should be relevant, and there are usually a large number of compounds that can be analyzed, and so it has to be scalable. Right? And as you will very well appreciate, human testing is not possible at an early stage. Okay? So what we need to do is something called as the in vitro assay. Okay? So the in vitro assay means it is usually a test that is carried out on isolated cells, tissues, enzymes, or receptors. You can also have animal models which are known as in vivo assays. Okay, so both of these are commonly used to test for new drugs. In general, in vitro assays are highly preferred over in vivo tests, okay, because it's much cheaper and it's much easier to carry out and less controversial, because animal studies are always associated with some controversy, and the in vitro tests can actually be automated. Okay. So therefore, in vitro assays are very highly preferred. However, in vivo tests are needed because sometimes when we have a lead compound, we need to be able to test whether it can show the desired activity in an animal. Furthermore, we need to be able to monitor the pharmacokinetic properties. Okay. So again, since we can't do these tests on humans, we need to be able to do this on appropriate animal models. So a variety of tests are usually carried out both in vitro and in vivo to determine not only whether the candidate drugs are acting at the desired target, but also whether they have activity at other undesired targets. So we need to achieve a good balance between the best activity and minimal side effects. So, something that really hits very hard at very low concentration the desired target may not always be useful, because that may have a number of other side effects which are not desirable. So, we need to be able to balance these two aspects. So, now let us look at some in vitro tests. As we discussed earlier, in vitro tests do not involve live animals, but instead they only have tissues, cells or enzymes. Okay. So, if we decide that we want to inhibit an enzyme, so let us say we have identified an enzyme that we want to target. Now, what we need to do is we need to be able to purify the enzyme. Okay. So, in the past this was a major problem, because it was very difficult to isolate and purify sufficient enzyme for us to carry out the test. But nowadays, because of genetic engineering, what people do is that they usually incorporate the gene which codes for the enzyme into fast growing cells such as yeast or bacteria, and then this enzyme is produced in large quantities, and this makes isolation quite easy. So, for example, the HIV enzyme HIV protease has been cloned and expressed in E. coli, and so once you have the purified enzyme, then you can use this enzyme to carry out tests. We will again look at some of these details later on in the course, but this forms a very good starting point to look for inhibition of an enzyme. So, developing a bioassay in vitro with an enzyme will involve purification of the enzyme, typically cloning and purification from a model organism such as E. coli, and then we need to be able to develop the experiment such that we can monitor for inhibition. Not only that, we have already discussed in detail about how to plot the Lineweaver-Burk plot, for example, and we can determine whether it is a competitive inhibition, non-competitive inhibition and so on. And of course, the important parameter that we wish to look at is the IC50, which is nothing but the inhibitory concentration 50%. Similarly, if we decide that receptor is a target, then receptor agonists and antagonists can be tested on isolated tissues or cells. So, these tissues or cells obviously will express the target receptor on their surface. So, what we would do is we would add the drug candidate to this tissue and look for activity, right. So, sometimes these tissues can be used to test drugs for physiological effects. So, for example, this bronchodilator dilatory activity can be tested by observing how well compounds inhibit the contraction of isolated tracheal smooth muscle. So, you can isolate this tracheal smooth muscle and figure out how well the compound acts in contracting it. Alternatively, the affinity of drugs for receptors, that is how well they can bind, can be measured by radioligand studies. So, we have already looked at this previously, and we have derived plots on this. Many in vitro tests have been designed by genetic engineering, where the gene encoding for a specific receptor is identified, cloned and expressed in fast dividing cells such as bacterial, yeast or tumors. So, for example, the Chinese hamster ovarian cells are very commonly used to overexpress some of the cloned receptors on their surface, and then we look for how the cell responds to it. In vitro studies on whole cells are useful because they are not associated with the complications that are typically found with in vivo studies. So, for example, when we are looking at an in vivo model, we need to figure out how to administer the drug, and this has to survive the metabolic enzymes and cross barriers such as the gut wall and so on. So, all of these can be somewhat bypassed when we are looking at in vitro studies. However, as you can imagine, in vitro studies also have very similar limitations, because we do not know whether the drug is going to reach the target of interest or not. But the environment surrounding the cells can be easily controlled. So, we can look at both intracellular as well as intercellular events, and it allows us to measure the efficacy and potency of the compound. There are also some things known as primary cell cultures, which are basically cells which have been isolated from animals, and these are not transformed or modified cell lines, and they can be produced from embryonic tissues or from other transformed cell lines which are derived from tumor tissue. Okay. So, cells which are grown in this fashion are all identical, and so they are very useful in testing out our compounds. So, if you are set out to develop antibacterial drugs, they can easily be tested by measuring how effectively they inhibit or kill bacterial cells in culture. So, what we can do is we can grow E. coli or staphylococci or any other bacteria in a safe environment, and then once they are in culture, then we can add our drug and measure how well they inhibit the growth of this bacterium. It may seem strange to describe this as an in vitro test, but since bacteria are actually living organisms, but just the fact that they are able to inhibit does not mean that it is going to work in an animal model. So, in vivo antibacterial tests are actually defined as those that are carried out on animals. So, there is a distinction here which we need to appreciate. Of course, the in vivo test can also be done on humans at a much later stage, and whether the compound is able to combat infection or not. In vitro tests are also used for understanding pharmacokinetic properties. So, for example, this Keiko-2 cell monolayer absorption model is used to assess how well a drug is likely to be absorbed from the gastrointestinal tract. So, this Keiko-2 cell is derived from colon cancer cells, and so this would be a good mimic of how the gastrointestinal tract functions and how well the drug is absorbed. There are also microsomes and hepatocytes which can be extracted from liver cells, and these contain cytochrome P450 enzymes, which we have discussed very extensively previously, and these can be used to assess the likely metabolism of drug candidates. And they are also used possibly to identify drug-drug interactions. So, another in vitro assay using artificial membranes has been developed to measure how well drugs will cross the blood-brain barrier. The next class of bioassays would be in vivo tests. So, in vivo tests, as we have looked at earlier, are on animals, and they often involve inducing a clinical condition in the animal, and then we observe for symptoms. So, the animal is then treated with the drug or drug candidate to see whether the problem is alleviated. So, this forms the foundation for in vivo tests. So, for example, inflammation is a huge problem, and so the development of non-steroidal anti-inflammatory drugs was carried out by inducing inflammation. So, this should be anti-inflammatory drugs was carried out by inducing inflammation on test animals and then testing drugs to see whether they relieve the inflammation. There are other animal models which we can develop, which are known as transgenic animals, and they are very often used. So, these are animals whose genetic code has been altered. So, it is possible to replace some of the mouse genes, for example, with human genes, and now the mouse produces the human receptor or enzyme in it, and this allows for in vivo testing against the target. So, if we have convincing data, in vitro data to show that our compound is hitting a particular target, then one would develop these transgenic mice and test them to see whether the target is really being affected or not. Alternatively, the mouse genes could be altered such that the animal becomes susceptible to a particular disease. So, for example, if we are able to induce breast cancer in animals, then we would be able to test the drug in a fairly relevant model. Of course, there are several problems associated with in vivo testing. It is slow, it is very expensive, and it also causes animal suffering, which is unacceptable to many. There are many problems associated with pharmacokinetics, for example. So, some of the results that we obtain from animal studies can be misleading and also difficult to rationalize if the in vivo tests are carried out in isolation. So, for example, if you get a negative result in vivo model and that the drug fails to bind its target, we do not know whether the drug has actually reached the target or whether it is not able to bind to the target. So, in vitro tests are usually carried out first to determine whether a drug interacts with its target, and then in vivo tests are carried out to test the pharmacokinetic properties. There are also cases where certain in vivo tests might be invalid. It is possible that the observed symptoms might be caused by a different physiological mechanism than the one intended. So, there were a number of promising anti-ulcer drugs which proved to be ineffective in clinical trials. Now, it is also possible that different results may be obtained in different animal species. So, the example that we can look at is this penicillin methyl ester prodrugs, which are hydrolyzed in mice or rats to produce the active antibacterial penicillin, but they are not hydrolyzed in rabbit, dogs or humans. So, if we have a case such as this, you would find that perhaps that the drug is not active in certain animal model, but active in other animal models. What is worse is that if we use this as a basis for human testing, and if the particular hydrolysis or activation does not occur in humans, then it would be a major issue. We also need to be able to look at the validity of testing. Okay. So, the testing procedures will have to be easy and clear cut. So, for example, when we are looking at antibacterial agents, we can easily test them in vitro by measuring how it can kill bacterial cells. We can also look at local anesthetic in vitro to look at how it's able to block action potentials in isolated nerve tissues. But there are a number of other cases where the testing procedure is actually more difficult. So, let us consider that you want to develop a new antipsychotic drug. So, what kind of an animal model would we develop? Will we be able to induce psychosis in an animal? All these are very complicated and probably impossible to solve, right? So, we would not be able to look at a simple in vivo test to test for antipsychotic drugs. So, what we would have to do is to work with the receptor or receptors which are involved in the medical condition. So, then we could carry out a battery of in vitro tests, and if these are in line with our expectation, then we would be able to administer these and test their efficacy in some model system, which is then taken forward. Of course, one problem with this approach is it is not always clear cut whether a specific receptor or enzyme is as important as one might think in the targeted disease. So, a lot of clinical data that is obtained suggests that there could be a hyperactivation of a receptor or depression of a receptor, for example, or a particular enzyme in terms of its expression levels. But this does not always translate to increased or decreased activity.